Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. In this episode of Careers in Discovery, I'm with Simon Young, VP of Translational Science at C4X Discovery. Now, Simon's career has taken some fascinating twists and turns, from veterinary medicine to electrical engineering, and eventually into diagnostics, drug discovery, and precision medicine. He shares how those experiences have shaped his approach today, and how staying flexible, taking on new challenges, and stepping out of your comfort zone have helped him to build an exciting career. Great to be back on Careers in Discovery this week with Simon Young of C4X Discovery. Simon, welcome to the show.
Tom, wonderful to be here. Thank you very much for inviting me.
Yeah, great to see you. And Simon, I'm really looking forward to getting into your career. I think there's been some fascinating twists and turns in there, and we'll come to those. You've joined C4X reasonably recently as the VP of Translational Research, Translational Science, and it'd be great to hear a little bit about what you're up to there. We had one of your sort of predecessors, Alan McCarthy, on the show almost 5 years ago now. I'm sure lots has changed, but it'd be great to hear about what you're up to at the moment.
Sure. Thanks very much, Tom. So I joined as VP of Translational Science, and one of the things that C4X Discovery is interested in is a precision medicine approach. We're in the inflammation and immunity space, and there's sort of a growing recognition that there is a real need to personalize some of the treatments for diseases such as rheumatoid arthritis or lupus. And so one of my roles is looking at how we can develop biomarkers, processes for identifying patients' response to therapies. And C4X has invested a lot in a genomic approach, which Alan McCarthy was setting up when he was there. I've now taken on his team, and we recently actually got a publication on this where we're looking at what are the genomic drivers of disease, and inflammation and immunity is a good area to apply that to, because there are strong genomic drivers and links for many of the diseases. So that's one of my roles, is to bring that forward and actually take that into the clinic. So develop ways of identifying patients who will respond to our therapies, who may have more advanced disease, or disease that is more likely to progress and requires a more aggressive treatment, things like this. So we're trying to personalize the treatments. And then the other part of my role, really, I bring some little bit of clinical experience. I've worked in AstraZeneca, as we'll talk about, and done some early and mid stage clinical development. And C4X Discovery has made a decision last year that it would move into early clinical development. Previously, C4X Discovery partnered out its chemical assets preclinically, but now we are going into the clinics, and that's a whole new area for the company. So I'm working with the VP of biology to lead us by the hand into the clinic and early clinical development.
Yeah, fantastic. So really exciting time then. And as you say, a lot of those foundations that have been laid at the business over the last several years now starting to progress into the clinic. It's an exciting, progress into the clinic under the company's own steam. It must be a really exciting time to be part of it.
It is very, very exciting. I'm really pleased that I can sort of bring, my expertise in, and I feel that I'm making a contribution to the company.
Yes.
And I think the other thing I notice is the tremendous excitement from the people there. Before, there's a lot of basic scientists, chemists, obviously, and biologists. And they're now seeing that their work is actually moving into the clinic and into patients. And everyone I talk to is really, really excited about this. It's a new area for them, something for them to learn, but also just to see the application of all their work.
Yeah, and it's interesting, isn't it? Because it's easy to forget that that's what you're working towards, even though that is always what you're working towards.
That's it. You can sort of get lost in the day-to-day minutiae, like any job, and you're there and you're knee deep in papers from Nature and so on, trying to look at the detail of biological mechanisms of rheumatoid arthritis or lupus or something like that.
Yes.
And then every now and then it's good to pop your head up and say, actually, what we're trying to do is make these patients' lives better.
Absolutely.
And we can actually do this. We can see that our compounds hopefully will do that.
Yeah, absolutely. No, well, we'll come back to that, and we'll come back to sort of your journey to that. And of course, precision medicine as a topic is one that's over the last few years been very central to your career. But it wasn't where you started. I was really interested looking into your background, Simon, that originally electrical engineering and veterinary medicine and a whole range of different things. So tell us a little bit about, I guess, where this career came from for you, and where engineering turned to medicine, turned to research, and how it all came together.
Yeah, it's a very long and very twisty pathway, and I sometimes pinch myself and say, "How on earth did I end up here?"
Yes.
Good thing is, I'm having a great time. So exactly. So when I was doing A levels, I was looking around for, okay, what would I like to have my career in? What should I study? And I've always been a scientist. I mean, I'm a scientist all my, ever since I was 8 or 7 or something, doing experiments on the kitchen table, so I knew it was going to be in science. And veterinary medicine appealed. At that time, it was a little different to what it is now. And it was more in the James Herriot era, where you could do it all yourself. And that really appealed to me at the time. So I applied for veterinary medicine. I think my second choice probably would have been chemical engineering. I just loved chemistry. But I applied for veterinary medicine and started that. And the course I was on, you could do in the third year, you could do an intercalated course really on anything that the university offered. And so I switched to electrical engineering and joined electrical engineers for year 3. And actually my first degree, because I sat exactly the same, is in electrical engineering. And I've always been fascinated by electronics. And I sort of grew up in the early days where we were just starting to get decent electronics, moving from the valve era into the transistor era, and also into the early computing. And I then did that, and then went on and did my clinical veterinary medicine. And then I started a PhD soon afterwards. Excuse me. And that's actually where I applied a lot of the electrical engineering. We were looking at how we can study how lungs work, lung mechanics. And this is very well-established field in people, but it's not very well-established in veterinary medicine. And animals have a number of respiratory diseases, particularly horses have a allergic type condition, which is quite prevalent and caused by dust and molds in the stables. But we're interested in objectively measuring horses' breathing. And of course, if you have your lung function measured, you'll be told to take a deep breath, blow out as hard as you can, and so on. Of course, you can say that to a horse, but nothing much happens. So there was this new technique which didn't require the animals to participate at all, and it was non-invasive and non-painful. But nobody had ever tried it in a large animal. And so that's what my PhD was on. We got it up and running. And actually, over the years, I've sort of semi-commercialized it, worked with a couple of commercial partners to produce the apparatus, which is being used in research. But then I got, because I was doing a PhD in the anesthetics department, I got interested in veterinary anesthesia, and then did further qualifications in that. And actually, my first job was in veterinary anesthesia, the specialist center in Newmarket. And I did mostly horse anesthetics for orthopedic procedures, and then small animal for oncology and ophthalmology. And then an opportunity came up in Canada to teach at the veterinary school there. And so I moved over to Canada, to Guelph, and taught at the Ontario Veterinary College, teaching veterinary anesthesia. But I've always been interested in science and research. I was running research programs on exercise medicine and lung function and so on. And it was okay, but I sort of, I looked at the academic path hard, and I wasn't so keen on it. So I thought, okay, I'll set up my own business, which was a mad idea. So I left the university, tried to set up my own business for a couple of years. It was disastrous. I didn't make any money. But I made a lot of contacts, and particularly with companies in the US that I was working with to develop new lung function systems. And then eventually we sort of ran out of money, and I had to get a job. And there was a place in the USA, which was then Shearing Plough, a pharmaceutical company, who I already worked with, supplying lung function equipment to them. And they wanted somebody to run some of their early programs. So we moved down to the USA. and I joined Shearing Plough, which is now Merck, and started in drug development. And that's what started me off in drug discovery. And quickly learned that I really like being in industry compared to academia. I like the goal-driven approach. You know what you've got to do. You've got to solve the problems. Not so good at blue sky research, where you think, well, it wouldn't be interesting to investigate this. I'm just not so good at it.
No. And I imagine that was a pretty good time to get involved with Shearing Plough, because I'm sort of casting my mind back to that period of time. And I guess very well-resourced company, you know, big enough to do stuff, but not one of the behemoths of the industry where everything's very corporate, always very research-driven. I guess they always had that link to animal research as well at Shearing Plough. That was always part of their sort of core.
That's right. Yeah. There was they had Shearing Animal Health, which was actually separate, but yes, that's right. And exactly that. They were very much the boom years for shearing because of loratadine, and that was their blockbuster, and they made a lot of money on that. And they did invest it in R&D, set up this fantastic place, new facilities in Kenilworth, New Jersey, and we're really looking around for the next targets and so on. So it was a real boom year, and because of their history in hay fever and asthma, they were investing a lot in the area, and that's what I knew about was lung function and lung diseases. So it was a super time to be there. There was, I said, it was well funded and lots of interesting research going on. And then we sort of thought about it hard, and we struggled a little to really find our feet in the USA. So we were looking to move back to Europe, and I looked at various positions, and a position came up in AstraZeneca.
Yes.
And what was their respiratory group, which was then based in Loughborough in the UK, with the Twin Centre in Sweden, in Lund. And there were two laboratories, both doing respiratory work. And so I moved to the UK from the USA, and started working there, both in the preclinical area, and then also started doing a bit of drug development work, working in the early phase 1 trials as well. And it's a long, twisty tale, because AstraZeneca consolidated and moved all the respiratory work, as you know, from Lund and Loughborough up to Gothenburg, where it still is. And we looked at moving to Sweden, and we thought about it very hard. It looked very attractive, but in the end, we got young children by then, and we decided we'd take them through the UK schooling system. So we then moved up to Audley Park, the big UK R&D centre, and that's one, I was talking to you from Cheshire, which is why we moved up here, and had a good time there. And then the AstraZeneca set up its precision medicine group and wanted to get a precision medicine, and I joined that group. I ran the imaging group for about a year and a half in between. So there's got quite a strong imaging facility, preclinical animal imaging with MRI and PET and SPECT and so on. And then AZ decided to set up a precision medicine department, which I joined. Initially, obviously, most of precision medicine, particularly at that time, was in oncology. So 80-90% of what we did was in oncology. So for 2 years, I worked on diagnostics for lung cancer and learned an awful lot about how you develop them, what a FISH test is, and set up laboratories in China and in the USA to enable phase 1 and phase 2A trials, because we were screening patients for genetic mutations in order to go on to some of our treatments. It was a really interesting time. The whole department was learning. We hadn't done it before, but we got several companion diagnostics approved. And then I sort of moved away from the oncology side, stayed in the same department, and we were looking at biomarkers for cardiovascular and respiratory disease.
Yes, I see.
Which is much, much, much harder. Oncology, my colleagues always hate me when I say this, but it's relatively easy to do biomarker and precision medicine in oncology, because it's driven by a driver mutation. What's driving the disease. And we know and it's a single point mutation, or it's a number of mutations in one gene, but it's relatively easy to measure that and develop a diagnostic, and the path is well trodden now. What we were trying to do is find out we have right back at the start, what's the biomarkers driving things like severe asthma, for example.
I see.
And so one of the areas I worked in there was in severe asthma, and we're developing the antibodies against IL-5, and AstraZeneca was developing one, GSK had got one as well, and we knew this was tightly linked to the level of eosinophils in the blood sample of the patient, and the response to the antibodies increased as you had more eosinophils. And we were looking, are there better biomarkers? So we started looking at some of the proteins produced by eosinophils, and to try to develop some quick tests, rather like the COVID test, the lateral flow test, that would give you a quick answer on what your blood eosinophil-derived neurotoxin, EDN, is what we were looking at. And it worked okay. We developed some prototypes. We put it into clinical trials. I think the company's still working on it. The idea was that it might be a better biomarker because it shows the activation of the eosinophils rather than just their presence. So these are active, what they call angry eosinophils, which are the ones doing the damage.
Yeah, I see. I see. And it's interesting, before we sort of go on to the rest of your career, I guess there's a few things there that I really wanted to explore. And I guess there's probably a few parts to this question. It's sort of forming in my head as I ask it. Because of the variety you've had in your background, I'm thinking particularly, the year you spent studying engineering, the time you spent with animal health, and then the focus that you've had for part of your career on diagnostics as well as therapeutics. I'm interested in how those things have perhaps influenced your approach to therapeutic research. And my assumption would be, and please do correct me on this, the engineering part, although it was a short period of time, teaches you a certain way of thinking about process and systems and things like that. One of the interesting things about animal health, I think, is that someone said this to me recently, and I can't remember who it was or what the context was, but they said the thing you've got to understand is that when you're dealing with different animals, you have to look at the biological principles, not that, a dog is not just a big rat. There's different ways that their body works, so you have to go deeper into the biology. And then also, I suppose, with precision medicine as a whole concept, it is that marrying of diagnostic and therapeutic, right? Because the diagnostic element is so important to effectively doing precision medicine. So there's a lot in that question. I apologize for dumping it all on you at once, but I guess my question is, how do you think that variety in your background has influenced how you think about the work you're doing now?
Yeah, and I think what it comes down to is, I said, with the engineering background, I became very interested in measuring things, physiological measurement, non-invasively, and that's what my PhD was on measuring mechanical properties of the lung non-invasively. And there's a lot of the analysis of the data there. There's quite a lot of maths in it and engineering principles. And I think I've sort of carried that. I've looked a lot over the years at the things like, I don't know, the Apple Watch and how people have had the sensors. And it's a very, very tough area to develop really good biomarkers. But I think I used to think that would be the way forward. I'm not so sure now because I think it's very tough. I think it's become a niche area. But we've seen, FDA approved diagnostics for atrial fibrillation on the Apple Watch and so on. So there are opportunities there. And I always thought, well, perhaps if, I was working in COPD for a while, patients with COPD who really, as you know, struggle to breathe and can't walk very far, and you think, okay, should we, put a smartphone in their pocket and measure their ambulatory activity, and that will tell whether they get they're getting better or getting worse, or perhaps having a flare of their disease. And there was quite a lot of interest in that. I think the problem is that it's very noisy data, and if you average across a large number of people, you can get strong insights and say, these patients, as they come up to a flare, they become less active and they sit in the house, and we can predict quite well when a patient's going to have a flare. But it works on a population basis. Trying to predict with good accuracy when I or one of my I'm going to have a flare is much more difficult.
Yes,
because that's on the individual, and you've still got very noisy data. And I think that's one of the big lessons I've learned is when you look at the big data and the results that you can get from that, it's absolutely fantastic science and it's great, but it's noisy. And then you try and translate that to a single person.
Yes.
And that's sort of where I think lab data is still very different to this population data. So I'm sorry, I'm rambling a bit, but I'm interested in the numbers. I start to focus on the numbers and precision. And this is what, if you go to the regulatory authorities with a diagnostic, they will focus absolutely on the numbers, the accuracy, the precision, and so on. What's the false positive rate, false negative rate, and so on. And so quite a lot of my work is about establishing those numbers and seeing how well a particular diagnostic is going to work. And it's quite difficult. We'll come to it. I worked in another company where a whole world was about how do you develop an assay that has got a very low variance, CV of 1 or 2%, something like that, very precise assays. Because if you send a blood sample off from you to the lab, it comes back with a CRP level of X, that the doctor needs to know that is a very precise measurement.
Yeah,
and I think that's what that's I think the sort of one of the two areas where we haven't quite married yet. We've got all this big data that's a bit noisy, electronic health records. I work with those. They give you insights, but translating that to a diagnostic that will say, yes, you should go on this drug, and I have a really high confidence of that, or you shouldn't, is difficult. Yeah.
Right. Yeah, absolutely.
But I think what fascinates me still is we're still measuring things, measuring things on people. We're seeing how we can analyze them. So I think I've gone on from sensors and lung function to what other data can we get on patients, and what can we measure? Ideally non-invasively, but it's usually unfortunately a blood sample. And then how do we analyze that?
Yeah, and I suppose you've touched on this a little bit, but one of the key challenges is really making sure that we're identifying the actual cause of the disease, right? Rather than a correlated signal.
Yes. And in many cases, that's not obviously known. And in inflammation immunity, there I'm working now, we know that there are strong genomic links to it. It's very but that's not everything. That's 40, 50%, something like that. The rest is environment and other factors that we don't know about as well. So we say, okay, what's the cause of rheumatoid arthritis? We don't know. We can say it's characterized by autoantibodies and so on, or lupus has got anti-DNA molecules. But what's gone wrong with the immune system, or what has pushed it that way? We don't quite know. We just can observe it. I do think we've made enormous strides in immunology over the last few years. We're understanding our own immunology so much better than we were 20 years ago when I joined AZ. So I think we're heading in the right direction. But what the cause is multifactorial, and the question is how much does each factor influence it, and that's what we're trying to decide. Yeah.
No, of course. And so this step sort of more formally into precision medicine started at AstraZeneca, and then through your work at Cymerus, and obviously with C4X as you say now, with Sysmex, sorry. Really interested in hearing about that part of the journey, because that's kind of where we left it. But also, I guess, your thoughts on precision medicine, where it is, because it's a concept that's been around for a little while now. I think sometimes misunderstood. Definitely been progress made, but hard to sometimes tell how much progress, and because it's a difficult problem to solve, right?
Yeah, that's right. So perhaps I'll start with the precision medicine, and I'll talk about Sysmex. So precision medicine, like many terms, is overused, and now it sort of means anything. But I think that the main thing is that we want to try and get the best treatment for an individual patient.
Right.
And if you take a disease like rheumatoid arthritis, there's a large number of potential treatments available. And what we want to do is get a particular patient on the right treatment first time. And I saw this particularly when I was working in asthma, is that it was very much a trial by therapy. So if you go in and the doctor says, okay, I think you have mild asthma, you'll get put on beta-2 agonists and possibly a steroid. And they'll say, see how you go. And you come back in 3 months' time and you say, how are you doing? And you may be getting better, or it may not be working. And then you say, okay, well, we might switch or change the therapy, try it for another 3 months. And that sadly, if you have patients who don't respond to the standard therapies, they can be a year or two of largely failed therapy. Obviously, a lot of patients, with mild asthma, they're fine. You go away. They've got a couple of inhalers, and they can learn to manage their disease, and they're absolutely fine. But I think what all the medics that we talk to and the experts say is, I would like to get the right treatment first time for the patient. I think that's what we're aiming for. And then what another arm, so that's the sort of general concept, and that's really good. And I think we're all striving towards that just by saying, okay, what information can we get about this patient? How can we dissect their disease? Particularly, are there different facets of disease? So we call it rheumatoid arthritis. Are there different drivers that give the same clinical picture? And I'm sure there are. The same in asthma. There are different designs, there are different flavors of asthma, who of course we recognize clinically that may have different drivers. That's difficult. Why do some people get asthma when they're 12, wheeze, but grow out of it 25, whereas women in their 40s suddenly go down with severe asthma that's not treatable by the, we don't, short answer is we really don't know. And it's not, there may be some genomic links and we looked at those, but that's not telling us the answer. And so I don't think we know. And I think, are they different forms of the same disease, or what is it? Or are they just different immune disorders that we called asthma because they manifest in wheezing and in a poor breathing? And I don't know. I think that's one area of precision medicine. What is the type of disease? And then the area I think that we associated perhaps a little bit more is if we have a novel treatment, and this goes back to cancer that I was talking about earlier, is where we know what the driving mutation is. And one of the big successes in cancer therapy over the last 10, 15 years has been developing specific inhibitors for those overactive genes. So obviously classically starts off with EGFRM in lung cancer.
Yes.
And there we can say, okay, this mutation, this gene has gone wrong, it's turned on permanently, it's driving the tissue growth, and we can identify it, we can stop it. And that's really, that's been a fantastic success story. And what I'd like to do is translate that into other areas. So if we say, okay, we have a particular disease mechanism that we're targeting. So if we take one of our therapies, for example, we're working on alpha-4 beta-7 integrins. And one of the questions is, okay, which patients are going to respond to those? And that's my current area of work. How do we identify those patients? And what's the spectrum? What's the spread of response in patients? And I think that's the sort of the second part of it that we tend to think about more when we talk about pharmaceuticals. And of course, if the link is very, very strong, then it becomes, you usually have a companion diagnostic. And if you look at the companion diagnostics, and to explain to the listeners, the companion diagnostic is defined as being essential for the safe and effective use of the drug. Okay. So before you prescribe the drug, you have to do the diagnostic test.
Yes.
And again, this comes from cancer because if you don't have the driver mutation, then the drug will not work. And they still have side effects. So actually, all you're doing is harming the patient rather.
Right.
Nearly all the companion diagnostics are coupled to cancer therapies. There are a couple which aren't. And I think the pattern's different in respiratory and inflammation immunity because in general, the drugs are much, much safer.
Yes.
The side effects are very much lower. So we can actually say, okay, try it and see how it works.
Yeah.
And so I don't think we need, I think there'll be fewer companion diagnostics where you have to do the test, but the test will guide the physician saying, okay, the likelihood of this patient responding to this treatment is high or low. And there is some diagnostics already been approved, for example, in RA in that area. And we're working in it for our new alpha-4 beta-7 integrin inhibitor. Yeah. And so where did I get to? So that's my thoughts on precision medicine. Is that everybody wants it. It's not easy outside of cancer. I think we're making progress there. One of the sad things I think is everybody wants it, but the commercial returns on selling the diagnostic are very, very low.
Right, I see.
And so I'll introduce Sysmex here. So I was working at AstraZeneca in the precision medicine group, and then an opportunity came up to set up a research and development lab for Sysmex in Cambridge. They wanted a branch. And Sysmex is a Japanese company that makes large automated diagnostic analyzers. Yes. Typically goes into hospital laboratories. And they have an R&D base in Kobe in Japan. But they also had small regional R&D labs around the world. There's one in Germany, the USA, Singapore, and then I set up the one in Cambridge, and it was absolutely wonderful setting. It was had great fun setting it up, working in Oxford Gene Technology, which sort of hosted us. It was a very weird setup. But we've got lovely labs and set it all up, got the instruments, and we were developing the nuts and bolts of the diagnostics and new assays to go on the automated Sysmex machines.
Yes.
And then I was working on particularly an introduction for a blood test for Alzheimer's disease to say, should you go on to the amyloid antibodies? So that was I did that for a few years, brought in a new team, trained them up and so on. And that was really taught me about the nitty gritty of the diagnostic side, what it's like to work for a diagnostic company, what's involved in the regulatory side, and what's involved in developing the diagnostic and doing all the validation and so on. And then I was looking around, I think, for the next challenge, and I saw actually one of my ex-AZ colleagues posted, I think, on LinkedIn. I think they'd been looking for a year and a half for a VP of Translational Science, nowhere, completely unaware of it. And she posted it on LinkedIn, and just happened to browse across it on I'd had a bad day or something. Browse it. Why the hell not? So I rang her up. Yeah. And and it was a sort of perfect fit. I just looked at the brief for the job, and I thought I've never fitted a job brief quite so well. So and the rest is history. So I then joined C4X Discovery in March.
Yes,
and where I can bring to the company is both the drug development experience, but also the expertise in diagnostic development and getting it through the regulatory process as well, which we're going to need in order to take our genomic diagnostics forward.
No, absolutely. And I think it's interesting, isn't it? I think it was a commencement speech that Steve Jobs gave, that he talked about you can only sometimes join the dots in hindsight. You can't always join them looking forward. And I guess this is where you've ended up. Yeah.
Well, yes, exactly. And I think that applies to our research or most research. I sit down at the desk and just stare at a blank screen because I've been asked to say, well, what's the strategy for our diagnostic for the next 6 months or something? It's incredibly difficult to predict the future.
Yes.
Particularly, you can, doing what everybody else has done is easy. Doing something new is extremely difficult. And so what I keep telling the team is nobody's ever developed this diagnostic before.
Yes.
And it's going to be difficult. And, whereas if you're copying what somebody else has done, then the path has been laid and you can follow it with a few minor tweaks. But I always say to the team, making something that nobody has ever made before is really, really difficult, because you've got to make all the mistakes, do all the thinking and so on, bring in all your scientific expertise. Steve Jobs is one of my heroes. He wasn't the nicest guy, but he created things that nobody else had ever created before. And he had fantastic vision. He really could. He could go to the engineers and say, "What do you think about this, Steve?" He said, "Yeah, that's okay, but it's not sexy enough. I don't like those buttons. I don't like that. It's not easy enough for the user to use." And he'd come in at this high level and say, "What I want is this. I want a phone they can hold in their hand. I don't want I don't want any buttons on it. I want them to look at it and it logs in." Tremendous vision.
Yes. Yeah, and I think it's easy to when you're in biotech every day. It's easy to underestimate the complexity of what you're trying to do. And we had Alistair Henry on the podcast recently, who's the CSO at UCB, and he was saying that whenever he has to explain to people who are not researchers what his team do and why it's so difficult, and also I think he mentioned why they get paid as much as they do, he explains to them that, well, I'm asking them to create molecules that have never existed to have an impact on things that we don't fully understand. So it's a bit tricky.
Yes, that's exactly right. Yes. Yeah.
Fantastic. So through the, as you described it, the twisty path that you've taken, what, from a career point of view, Simon, what would be the key things that you'd sort of draw out as, the formative experiences or the most important lessons you've learned along the way, that sort of stuff?
Okay. So I think the lesson, most important lesson I've learned, and this is what I say to everybody I interview who when they're coming for a position in pharma, most of them, of course, from academia or with a PhD or something, I say to them, the one thing you have to be in industry is flexible, because what you're working on now, you will not be working on in 6 months' time.
Yes.
So you may think your PhD subject is the only subject worth thinking about, but actually in 6 months' time, you'll be working on something else. So that's my biggest advice to anybody thinking about, should I go into industry? The science is great. It's, industry has really made, pharma industry has made huge strides forward, improved its ethics, cleaned up its house. It's a great place to do science, but you've got to be flexible. If you say, I'm only interested in X, just stay in academia, and write your grants, and that's fine. I think that's the biggest lesson I have for other people, and that's just because of the nature of industry. I love it, new challenges, new things coming along, and so if people say, well, how on earth do I do that? I don't know about that. That's it. Okay, it's going to be lifelong learning, and what I want you to do is apply good scientific principles and try and bring in as much outside information as possible. So, how are we going to solve this problem? How are we going to create this molecule? Okay, well, we don't know. We don't know what this molecule is going to look like, but we can say, okay, we know chemical principles. We've got the crystallography structure. We can sort of make it in by a guess, and that's how you do it. And you bring in, and if you've got chemical challenges, you say, okay, what's this reaction? How do we make this bit here? And what's everyone else done? And it's the same in biology. Is what are we looking for? What are the genomic drivers of this disease? Okay, well, how could we perhaps deduce that from the patient records and so on?
Yes.
And then so it's a lot of it's about scientific principles and just general reading. Yeah, it's that's my one thing. I think. What have I learned? I think it took me until about 2 years ago to think, what do I really want in a job? And so this is personal to me.
Okay.
Everyone says, I talk to my children, and you say, what do you want to be doing in 2 or 3 years' time or 5 years' time? My daughter had an interview for her first job yesterday, and I was helping her last week with some prep, and I said, they're going to ask you, what's your vision? What do you want to be? Where do you want to be in 2 years' time, 5 years' time? And of course they did. And everybody does. It's very difficult. I think I finally realized what the 3 drivers are. And for me, this is a personal one. Is I've got to be helping people. The first one is important. I'm helping people. Second one is that it's based on science.
Right.
And the third one is that there is lifelong learning. So I've got to be learning new stuff. I've got to be in science. And I've got to be helping people. And that's why I've ended up in pharmaceuticals.
Yes. And of course, if you can do that in your career, that then opens up a lot of options to you, doesn't it? Because I think people do get very blinkered in what they do, especially in science, because what you do is so specialist a lot of the time. As you said, if you just want to go down this one path and learn more and more, you could do that, and there's nothing wrong with that. And actually, that's really important. We need people to do that.
Yeah.
But as you say, if you then break that down to real first principles of, well, these are actually the things that are important to me, well, then it doesn't matter if you're in this disease area or working on this mechanism or whatever it might be. If you can still meet those core criteria that you have in lots of different ways. Which opens a lot of doors.
Yeah, that's right. And I think it's worth taking on new challenges and going to different areas. Otherwise, your CV can become very narrow. And I found this out the hard way when I'd worked in veterinary medicine. I'd done research on lung function. I was specialized in anesthetics, veterinary anesthetics. And then you go to a pharmaceutical company and they say, we don't need any of that. We're not a veterinary pharmaceutical company. And it was when I was looking around for a job in Canada, it was a real eye opener. Is that I thought, okay, I've got rather a narrow CV. And so I think when you're changing direction, it can be painful at the start, but at the end, when you've got experience in different areas, and of course pick up some management experience and things on the way, then it opens lots and lots of doors.
Yeah.
Whereas if you only looked in this one narrow area, have no expertise outside that, okay, you're only going to find a job in that area. So I think that in terms of flexibility for the future proofing, it's important to move into different areas. I've got some of my team at Sysmex have strong molecular biology background, but they need to move beyond that and use, okay, I've got that now. What else am I going to learn? Protein chemistry, things on protein biochemistry and so on. Yeah.
And as you say, it's hard to predict in advance, but eventually you find yourself as the only person qualified for a position that's been open for 18 months that's really key to a business, and serendipity sends it your way, and those things line up. But you can't necessarily plan those things, but it's the breadth of experience that allows you to find those opportunities, I suppose. And
I've sort of I've not quite blown with the wind, but I've sort of never really said I want to be, from in 10 years' time, I want to be CEO of a small biotech. So I've never done that. I just say, well, that looks interesting, okay, I'll do that for a bit and so on. And I'm bit fed up with what I'm doing now. What else is there? So I've sort of twisted and turned, partly because I've been a bit opportunistic rather than sort of focusing on the 10-year plan. And I think so. It's been good. Yeah.
And what this may be a very difficult question to answer, and it's not one I've prepared you for, so I apologize. But what do you think has allowed you to twist or turn forwards? Because I think a lot of people flip between different things and don't make a lot of progress. Whereas, if you look at your career, you've consistently progressed in terms of sort of the level of roles you've been in and things like that. Do you think there's anything that has helped you to do that?
That's a really good question, Tom. And as I'm thinking, and I think it's, don't be afraid to tackle new challenges.
Right.
And we had this phrase at AstraZeneca from the CEO. They had these various company mantras, which were really well thought out, actually. And one of them was do the right thing.
Right.
By that, it means it's very easy to do what you know how to do. So if you've got a problem, biological problem, well, I know how to measure a molecular biologist and do that. Because it's a comfortable area, you're comfortable, you know how to do it. But if we don't need that approach, what we need is protein biochemistry,
right? And
none of us know anything about protein biochemistry. Okay, how are we going to tackle that problem?
Yes.
And I think, and that certainly helped a lot at AZ when, what are we going to do with this drug, or how are we going to develop this drug? Well, what's the right thing to do? We should be aiming to have a clinical trial in this type of patient or something, or we need this physical chemical property. We haven't got it. We haven't been able to do it, but we need it. And I think that just don't be afraid to tackle the unknown problems. So sit down and say, okay, what would make the biggest impact to this project? What would move the company or the area of research, or what would make the biggest impact for patients? This isn't, I'd like to solve RA. This is, okay, on the small level, but what would the best properties of this drug? If it's, would it be a once a day drug, a twice a day drug, an injectable, an inhaled drug? What side effects are the worst for the patient? And so, and think about, those are the problems. Those are the things which will really make a difference. How do we tackle those? Not, okay, I can measure its effect in some assay, on the biological assay, and we can improve on that. If that's not what's needed, don't do it.
And I think
so. And I think that's it. It's essentially, and I think the other thing is you want to be outside your comfort zone for about 30% of the time. So push yourself outside your comfort zone, and there's lots of business what they call them, ideas about this, and you can go on business training courses. But basically, if you're inside your comfort zone, your world shrinks.
Yes.
Because you stay more and more inside your comfort zone, so you don't expand. If you're way outside your comfort zone, you're overstressed.
Yes.
But you should be about roughly a third of the time, you should be feeling slightly uncomfortable, thinking, I don't know how to do this.
Yeah.
And that will get you thinking about it. And then at night, or when you're driving home or having dinner, you'll be going on in your mind. You think, oh, what about trying this? So I think that's it. Yeah, push yourself outside your comfort zone for a bit.
Yeah. So get to the core of the question that you're trying to answer and push yourself outside your comfort zone. And I was having a conversation with someone in some LinkedIn comments, I think it was yesterday, about this, that if you're taking on something, whether it's a new opportunity or a project or whatever, if it doesn't make you a little bit nervous, not completely terrified, but a little bit nervous, you're probably not pushing far enough.
I'd agree completely with that. Yes, absolutely. Yeah.
Fab. Well, Simon, there's lots of other things I could ask you about, but we're out of time, unfortunately. It's been fantastic to speak with you. Thank you so much for taking the time to join us.
Thanks very much, Tom. It's been wonderful talking to you.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.